Staphylococcus is one of the gram-positive cocci of medical importance. The common Staphylococcus species are Staphylococcus aureus, Staphylococcus epidermidis, and Staphylococcus saprophyticus. Of these three, Staphylococcus aureus is the one that is considered pathogenic. As you begin the identification process of any organism, you always start with the microscopic morphology. In this case, Staphylococcus is gram-positive cocci. Once you know you're working with gram-positive cocci, there's a very short list of organisms you could possibly have: Staphylococcus, Streptococcus, or Enterococcus. The next step is to look at the macroscopic morphology. On nutrient agar, Staphylococcus tends to grow as circular, entire, and convex colonies that are either white or cream-colored depending upon the species. On sheep blood agar, the colonies are also circular, entire, and convex, and white or cream-colored, and they may show beta hemolysis, depending upon the species. The next thing you do is your biochemical testing. For Staphylococcus, you'll need to do catalase, coagulase, mannitol salt to check for growth and fermentation, and novobiocin susceptibility. To do the catalase test, you place a drop of 3% hydrogen peroxide on a glass slide. You will then select a colony to be tested and mix the colony in a drop of hydrogen peroxide on the slide. Some organisms produce the enzyme catalase. This test will check for catalase production. Hydrogen peroxide in the presence of the enzyme catalase breaks down to water and oxygen. Oxygen will be released as bubbles. Bubbles will equal a positive test. That means catalase was produced by the organism. And no bubbles will be a negative test. Catalase was not produced by the organism. The catalase test is what we consider a spot test. You perform the test and get the results immediately. All Staphylococcus species are catalase positive. If your gram-positive coccus is catalase positive, the next test you want to do is the coagulase, and the coagulase is also a spot test. Some organisms produce the enzyme coagulase. Coagulase is an enzyme that will convert fibrinogen to fibrin. Pathogenic organisms produce coagulase because they can use this enzyme to coat themselves in the host fibrin. If they're coated in host fibrin, they basically can hide from the immune system and won't be attacked. The coagulase test checks to see if the organism produces the enzyme coagulase. It uses latex particles that are coated with fibrinogen. If coagulase is present, the latex particles will clump or agglutinate. You obtain a test card and place test latex in one circle on the card. This is the latex particles that are coated with fibrinogen, and you place control latex in the other circle on the test card. This is to make sure that agglutination is truly the result of coagulase and not some other activity. You mix a visible mass of cells in each drop. Now between each mixing, you sterilize the loop. So you pick up your mass of cells, mix it in one drop, sterilize the loop, pick up another mass of cells, and mix it in the other drop. You want to mix the organism and the reagent and spread that to cover the entire circle. You then gently rock the slide and observe for agglutination. Agglutination in test latex and not in the control is a positive test. So this little bit of graininess is the agglutination. There is no agglutination in your negative control. No agglutination in the test latex or control latex is a negative test. If both circles appear smooth, then you have a negative test. If there is agglutination in the negative control, the test is invalid. If you have a coagulase positive organism, you have Staphylococcus aureus. But coagulase negative organisms will need more testing. These tests will require incubation. You will need to do the mannitol salt test and the novobiocin susceptibility. Mannitol salt comes as an agar slant, so all you need to do is streak the slant with the test organism and then incubate the slant for 18 to 24 hours at 35 degrees centigrade. The novobiocin susceptibility test is a disc diffusion test. You are going to use nutrient agar, and this time you're going to streak the agar for confluent growth. Since you're going to be testing 3 organisms, you will use the pen and divide the plate on the back into thirds. And then with the test organism, you will streak each quadrant for confluent growth. You don't want any isolated colonies. You streak as close together as you can because you want what's called a lawn of organism. Then you place a novabiocin disc in the center of the lawn and gently press the disc to the agar using either a sterile loop or some heat sterilized forceps. You will incubate the plate for 18 to 24 hours at 35 degrees centigrade. Like any other agar plate, this is incubated agar side up. After incubation, you will look at your mannitol salt slant. There are actually two tests in this particular slant. Mannitol salt contains salt which inhibits growth of some organisms. So you'll want to record whether there is growth or no growth on the slant. It also contains the sugar mannitol. Some organisms can metabolize mannitol to acid end products. This agar is red because it has phenol red in it. Phenol red is a pH indicator. At neutral pH, it is red. If the agar becomes acid as a result of acid end products, the agar will turn yellow. So looking at growth, you want to check your slant for growth. Pay no attention to color at this point. Both of these are positive for growth because we have growth on both slants. To look at the mannitol salt fermentation, you're now going to look at color. Yellow means that acid was produced. This is a mannitol positive organism. Red would mean no acid was produced, so this would be a mannitol negative organism. Here, the red one, this would be negative for mannitol fermentation. The yellow here would be positive for mannitol fermentation. After incubation, you will want to look at your novabiocin susceptibility plate. Novabiocin is an antibiotic, and that is what is in the disc that we put on the agar. The antibiotic will diffuse from the disc into the agar. If the organism is susceptible to novabiocin, it will not grow on the agar around the disc. You should get a zone of growth inhibition, or what we call a zone of inhibition, around the disc. You will use your metric ruler to measure the diameter of this zone, and you measure it in millimeters. That's the smallest unit on the metric side. If the zone diameter is greater than 17 millimeters, the organism is susceptible to novabiocin, and you use a capital S to record that test. If the zone is less than 17 millimeters in diameter, then the organism is resistant. You use a capital R to record the result of that test. So here you see a zone. All of this is where there is no growth. So going across the diameter, this is about 24 millimeters. That's greater than 17, so this is reported S for susceptible. This one, there's virtually no zone of inhibition. Less than 17 millimeters would be recorded as R for resistant. To recap your test results, you had 3 organisms you tested: Staph aureus, Staph epidermidis, and Staph saprophyticus. All Staphylococcus species are positive for catalase. Only Staphylococcus aureus is positive for coagulase. All Staphylococcus species tolerate salt, and so they will grow on mannitol salt agar. But only Staph aureus and Staph saprophyticus ferment mannitol. Only Staph saprophyticus is resistant to novabiocin. The other 2 species are susceptible.